Okay, this this little lecture accompanies Chapter 12 on DNA technology, and probably the greatest revolution in biology has been about recombinant DNA. And so these are the most important applications, money wise, of new DNA technologies, and they've led to really lots of new products. And so we're going to go into that. And these are just techniques for moving genes from one organism to another that have been so valuable. And so recombinant DNA just means DNA from two sources. And so, since all cells use similar mechanisms to handle DNA, a gene that's moved from one cell into another organism, a completely different organism, will produce the same protein. And so that allows us to move traits from a bacteria to a plant, from a human to a bacteria, and back again. And it's really, you'd think that this is really technical and difficult, but it's not. It's easy to do. It requires that you know where you want DNA. You know that there's a gene of interest in a particular organism, so that's going to be your source DNA. And where you're going to put that into is DNA from a phage or a plasmid, which you can move into other cells. And so you get those two types of DNA and you cut them with a restriction enzyme. Okay, and so you cut them with the restriction enzymes, and then you mix the two types of DNA together. Both have been cut by the same restriction enzyme, and then you add another enzyme, DNA ligase. And that DNA ligase will splice together, glue together those different pieces of DNA. And that then you'll have your gene of interest in your plasmid or your phage, and then you can put that into a host cell, and the host cell will start producing proteins, the proteins that you wanted. Okay, and so and there's details that I'm not going over here. You have to kind of sort things out. Sometimes you'll get multiple copies, sometimes you'll get 0 copies of the gene you want in the phage, but there are techniques for sorting that out. But what it means is, say you have a gene for a human hormone that you want to be able to produce that hormone in unlimited amounts. Okay, you can, knowing what cells produce that hormone, you can take those cells and cut them with an enzyme, and then you can put that gene into a bacteria. Okay, that bacteria then will make the product of that human gene. And so you'll have unlimited amounts. And in fact, the first really big application of this was a medical application, and it was using insulin to treat diabetes. And so we took a cell from the pancreas of humans, cut it with an enzyme, a restriction enzyme, and then we cut a plasmid with that same restriction enzyme and mixed them together with DNA ligase. Okay, and then that plasmid was a recombinant. It had the human gene in the bacterial plasmid, and that could easily be inserted into a bacterial cell. You can then grow that cell in a fermentation tank, in a very large test tube essentially. And every time that cell is growing, it'll pass on to its daughter cells a copy of that human gene, and all those cells will make insulin. And that insulin can easily be purified. And all it takes to get more of that insulin is to feed that bacteria sugar. Okay, it was remarkable. It replaced a really laborious process. And there's a little link for 2 tons of insulin from pig pancreases. But we can do this to get clotting factors to treat hemophilia, blood clotting disorder. We can make growth hormone to treat dwarfism, or using it for sports for illegal performance. We can get cancer treatment drugs called interferon. We can make vaccines. There's unlimited medical uses of this. Okay, even more important have been some of the agricultural uses. We've cut a gene from a bacteria, from Bacillus thuringiensis. That gene is for a toxin that attacks bacteria, and we've put that gene in corn. And now corn makes a toxin that kills insects. has no effect on humans at all, but it kills insects. The corn is, in fact, making its own insecticide. Really remarkable. And so farmers don't have to spray all sorts of insecticide on the corn. They don't have to go over plowing over and over again. They don't have to spray as much fungicide, and the corn comes out looking beautiful. Another important agricultural application is a gene for resistance to a very broad spectrum herbicide called Roundup. These soybeans have unlimited resistance to Roundup, and Roundup kills virtually all broadleaf weeds. It's really valuable. There's many other applications as well. We can make unlimited amounts of enzymes. We can make bacteria that are good at breaking down waste products or even cleaning up oil spills or toxins. Um, we used to make stone wash jeans that were actually washed with pumice stones to make the cotton softer and easier to wear. But now that's done with an enzyme that's made from biotechnology. So this has really been a remarkable advance worth billions and billions and billions of dollars. Okay, so in summary, DNA is the same in chemical in all cells. It has the same function in all cells, and all cells use the same code for making proteins. We have enzymes that can cut and paste DNA to allow us to make recombinant DNA. And then we can insert that DNA into cells using plasmids and phages. We can make medications, ag products, lots of things. We can treat disease worth billions and billions of dollars, and it's not that difficult to do. So, okay, that's the end of that program.